Good evening, ladies and gentlemen. Today I will be talking about biofuels from algae and microorganisms. This is the important case which I developed based on literature. Why? Because there are few time to replace our, let's call, traditional energy source and material source as fossil fuels. And due to the depletion that almost 80% is burned. And 2020, in 4 years, oil will be only on Middle East Asia. And in 50 years, there is no oil, further, no gas, no coal. Therefore, we should stay how to recycle. And all of us, we still produce organic waste, also oil from organisms. So we need to accelerate process to make suitable this waste to become the fuel. Because oil, carbon, these are waste of biomass which will stay underground as per special conditions several million years, several 100 billion years. So we need to change and prepare biomass such that we obtain fuel. New organisms as fossil fuels can be cooled to plastics and carbon dioxide. Of course, plastic is also, after petroleum, can be used, but we are making here focus on microbial potential. So this carbon dioxide, which is used, can be used by plants and bacteria to make. Because organic waste coming from biosynthesis, applied farmland, applied fungi, applied animal, which can generate plant to biofuels, and fat and proteins comes from animal also and fungi, which can be source of polymers and also as a fat of biofuel. These polymers can be used to build material for maintaining and chambering the places of the oil production. We first talking about several points that were between 2018 and 2024 because we have to know that most started science mixtures beside catalysis of nitrocellulose in case of dark fermentation, which is transformation of substrate into hydrogen, by photolysis, which here we have the algae and microalgae, which can be produced photofermentation that works is anaerobic under only presence of light, and microbial electrolysis cell, which decompose organic matter due to the potential. And next, the last is anaerobic digestion. We need to find some started science mixture besides of aerosols. This is in 2018. Now, most combination are tried, and this is the find. In case of biophotolysis, is simply not so composed, so organic waste, just to produce biophotolysis and some farmers. Now, with a big fuss as being source after other microbials is important for is important for into hydrogen and as a part of mixed substrate in other techniques. And due to easily go extraction to other biofuels and polymers. Sugars are much modeling attempts were based on sugars. Now they allows to co-digestion for more hazardous ways as asbestos. In case of biophotolysis, sugars are potential wood killing attempts for algae. Now sugars are more efficient part, so more and more interested part of farmers. In case of photofermentation, there was less low interest, mostly as trial to of cooperation of dark fermentation with photofermentation, but now they extended much there because not only organic acids are sourced, but also glucose, sugar proteins, and this extension is that always can be be developed but for fermentation. In case of water splitting, only parts can be charged without charges, but now we are looking for other ways for develop help gaining energy for splitting. As part of Nicholas's waste were sugars, now a combining of import with the dark fermentation important for the upgrading of metal production. In case of fats, there are trials of checking pathways. Now fats are very good and even similar to glucose source of hydrogen and be processed with other techniques. In biophotolysis were very slow by before 2018. Now fats are by extraction have significant content of biodiesel and or biolubricants. An additional process after the is in case of become in case of photofermentation, which earlier was available. Though the fats are not still a substrate both for microbial fuel cell, this is the pathway to source search. In aerobic digestion, they used them for industry olive, and now they upgrading as well. In the proteins were very seldom only theoretical potential. Now change theory of the fermentation due to supersession. In case of biophotolysis, no proteins, but now these proteins are both source of polymers and pharmacy. Photofermentation doesn't take account as well as anaerobic digestion some interest, especially battery waste. Now, especially in Taiwan and Asia, are big focusing of how change because to make fertilizer and other techniques. In case of other, mostly batch, now are trial to make upgrading processes and mixing. Process trial photolysis hydrogen production with increasing of phytochemicals. Hydrogen production is additional to carbon dioxide capture and pharmacy and green polymer source. This method widely tested for different feed is part of combination of future waste utilization. They limited mostly to water started to intense substrate. Industrial attempt for ammonia splitting are common after discovery of Haber processes. In case of dark fermentation, we have main pathway traditional of polycarbohydrates, which go to sugars, acidogenesis, and finally metagenesis in case anaerobic. In case of dark fermentation, it stops on acidogenesis, so no further action. Therefore, we have then as example the conclusion. In case of relatively similar, just we have protein and sulfur, which we can check in such substance and prove that origins of hydrogen sulfide isn't from the substrate but from organic bacteria waste, so which is didn't consider. So this is a conclusion: the more simpler, the cheaper, the more hydrogen, but less less emergency utilization. So we need to focus pathway, but compose can be a source of polymers, and this is the hope. And thank you very much for listening to me.